I told you some terms that mitospore, those spores which are formed by mitosis are called mitospores, and mitocytes are those cells which undergo mitosis, and mitophase is the phase of the cycle or the life of an organism during which mitosis takes place. I even explained to you how a duplicated amount of DNA, a chromosome, attaches to the spindle fiber with the help of kinetochores, and the central portion is the primary constriction or the centromere. Then I gave you these graphical things regarding minutes, how many minutes are required, and I explained this much. Now let's start the concept now. Let's understand what exactly is estral mitosis. And what is estral mitosis? If during the process of mitosis esters develop on the both ends, it is called as estral. Ester means star-shaped structure, so it will be called as an estral mitosis. And because esters develop on both the poles, it can even be called as amphiestral mitosis. So animal karyokinesis, or you can say animal cell division, is generally called as amphiestral or estral mitosis because esters develop in the case of the animal cells, and very rare examples of the plant cells will be discussing. So esters develop because of the pair of centrioles which are present at both the poles. Anestral mitosis, so a mitosis in which ester is absent, is called as anestral mitosis, and example is the plant cells, because in plants you don't find esters developing on the pole, so it is called as anestral mitosis. Now another type of mitosis possible is pre-mitosis, or you can say it's the mitosis which came before the real mitosis came. It is also called as intranuclear mitosis. Now intranuclear means inside the nucleus. That means nuclear membrane does not disintegrate. So in the example, there are many protozoa. Example amoeba, yeast, fungi, some of the algae, where you will find the nuclear membrane will not disintegrate. That means the nuclear membrane will remain as such. It won't disappear by the end of the prophase. And then what will happen? Spindle will develop inside the nuclear membrane, and such a type of mitosis is called as intranuclear mitosis. Now another type is endomitosis. Now endomitosis is what? Endomitosis is also called as endopolyploidy or endoduplication. Reason, after S phase, the nucleus will never undergo spindle formation. That means what? Amount of DNA has been doubled, number of chromosomes have been changed, but there is no separation of the nuclei content. So nuclear content will remain as such. That means a 2n cell will change into 4n cell, or a 4n or maybe 4n cell change then into the 8n cell. It is very common among the liver cells, even in the case of the human body. So endomitosis occur in some liver cells, and where the haploidy number can be tetraploid. Remember, human body, normally general body cells which are called as somatic cells, they are diploid. Diploid means, diplo means 2, so 2n. That means every chromosome exists in two bodies. That means two. One has come from mother, that means the egg, and the other has come from father, that means the sperm. But in the case of the liver cells, the same number of chromosomes have increased. Reason is that they have undergone endomitosis or endopolyploidy or endoduplication. So in this case what happens is chromosome will replicate, but there is no nuclear division. As a result, the number of chromosomes, or you can say the genome size, is going to increase. So it is also called as polyploidy. Poly means more than 2 in this case. So liver cells can easily be found to carry tetraploidy or octaploidy. Tetra means 4n, octaploidy means 8n. Then there is another type which is called as the free nuclear division. Now what is free nuclear? Remember in the last chapter's lectures, I told you plasmodium in the case of slime molds, syncytium in the case of the animal cells, Cenocytic in the case of fungi and plant cells. Remember the concept in which a cell may carry large number of nuclei. So such a type of division where there is nucleus is only dividing, that means karyokinesis is going on, but cytokinesis does not take place. So the same cell will carry large number of nuclei, and this can be called as free nuclear division. If you need example, Rhizopus is an example from fungi, Wucheria from the algae, and if you need example from the plants, then endosperm in 12th class we are going to learn. Endosperm carry large number of nuclei, and it is called as free nuclear endosperm. You all enjoy drinking coconut water or coconut milk. So that coconut liquid is nothing but large number of nuclei. Now next is dinomitosis. Now what is dinomitosis? In the third chapter of the 11th class book, you are going to learn quite late now, but not now. Later in the 11th class, you are going to learn this chapter where you will have the topic of dinoflagellates. So there are dinoflagellate protista which show a different type of mitosis, which is called as dinomitosis. So what happens in dinomitosis? Chromosome, the genetic material always remain condensed. So whether it is interphase, because generally we believe interphase means genetic material is loose. But in the case of dinoflagellates which perform dinomitosis, even the non-dividing nucleus will carry chromosome, that means always condensed form. And then the next thing is nuclear envelope will remain as such, and spindle is not formed. So there are other type of channels which will develop, which are called special channels. These channels help in equal distribution of the genetic material. So what happens in dinoflagellate? Nuclear membrane as such, spindle does not develop, and the next thing is they will carry a chromosome. That means always a condensed type of structure is there. Remember, dinoflagellates are considered as mesokaryotes. There are two names you have learned so far: prokaryotes and eukaryotes. So dinoflagellates are mesokaryotes because their some of the qualities are like eukaryotes, but all qualities are not like eukaryotes, so they are called as mesokaryotes. Like spindle is not there, and nuclear membrane will not disintegrate, but chromosome is there, so that's why they are called as meso, means middle, somewhere in the middle between prokaryotes and the eukaryotes. I hope this is clear to you.